Today we are going to talk about the muscle development. We'll talk about the development of the skeletal muscle, cardiac muscle, and smooth muscle. Firstly, we start with the skeletal muscle. And from the historical uh, point of view, the skeletal muscle is striated muscle, multi-nucleated. The skeletal muscle cell is multi-nucleated. The myoblast firstly forms, then elongate. After elongation become fused, and this will result in multi-nucleated skeletal muscle cells or fibers. And then after this, the cytoplasmic myofibril is formed. As you see here, this is an H&E section of skeletal muscle. You notice the peripherally located nuclei. So it starts by formation of myoblasts that elongate and then fuse to form multinucleated cells, and then the formation of cytoplasmic myofibril. The second is the cardiac muscle, and the cardiac muscle cells, they are branched and fused, and there are what is called intercalated disc that found at horizontal or at horizontal and the horizontally located intercalated disc perpendicular to the axis of the cardiac muscle. How it forms? The cardiac muscle develops from the splanchnic mesoderm around the cardiac tube, and then the myoblasts adhere together by intercalated discs. Then after that, the cytoplasmic myofibril is formed. As you see here in this histological section, it's the longitudinal section through the cardiac muscle. You see the nucleus, and here the intercalated disc. The specialized muscle cells formed from the neural crest cells. They form the Purkinje fibers, which forms the conducting system of the heart. So, uh, third type of the muscle is a smooth muscle. As you see, it is a spindle-shaped smooth muscle cells with oval nuclei, centrally located nucleus. What is the origin of the smooth muscle? The smooth muscle in the aorta and the large arteries originate from lateral plate mesoderm and neural crest. The smooth muscle in the coronary arteries, they originate from proepicardial cells and the neural crest. Smooth muscle of the gut originate from the splanchnic layer of lateral plate mesoderm. And finally, there is an exception that the muscles of the pupil in the eye and the mammary gland and the sweat glands, they originate from ectoderm. Muscles in these structures in the pupil, muscles of the mammary gland, and muscles in smooth muscle in the sweat glands, they are formed from the ectoderm. Let's talk about the skeletal muscle, and that forms the head muscles, the axial muscles, and the appendicular muscles or limb muscles. The appendicular muscles, we mean the limb muscles. The axial muscles, muscles of the back, and muscles of the abdominal wall. We have two important structures, the somatomeres and the somites. The somatomeres, they are 7 and they are partially segmented. They contribute in head musculoskeletal development. As you see here in this uh, diagram or image, you see the neural tube in the middle, and here in the cranial region we have the somatomeres, which are 7 in numbers, and they are partially segmented, not completely segmented. They contribute in the development of the musculoskeletal system of the head region. Caudally, we have, this is the caudal orientation, and here caudal to the somatomeres we have the somites, and they are 42 to 44 in number. They are completely segmented. The somites starts from occipital to the tail bud. In this diagram, we have in this image, we have transverse section through embryo, and we have the here, what is this? The neural tube, and here the notochord. This is a somite, and this is the lateral plate mesoderm. Here, this is incipient frontier border that separates the paraaxial mesoderm or the somites from the lateral plate mesoderm, and it separates two domains of mesoderm: primaxial, which is in the green color, and the abaxial, this uh, red color or orange color. In B, this is a further development. Some of the cells from the somites cross the frontier and enter the region of the abaxial. And this is in C. Yes, part of the somites are involved into the abaxial domain. As we see the primaxial forms certain type of muscles, and the abaxial forms certain types of muscles, and we will explain this later. So we have the differentiation of muscles into dorsal group of muscles or the back muscles, and abdominal wall muscles or ventral body wall muscles, and the limb muscles. In a minute we will see which is the origin of each group of muscles. In A, this is the differentiation of the somite. We have mentioned this differentiation in previous lectures, but we will repeat it again for quick revision. In A, the image A, here we have the neural groove. The neural tube is not formed yet, and we have the notochord ventral to it, and we have the somite and the ventral somite wall. In B, the somite has further differentiated with further development into skeletal group of cells called the skeletal, and we have the dermatomyetome, which have dermatome part and dorsomedial muscle cells and ventrolateral muscle cells. In C, further differentiation occurs, further into skeletal and myotome. The dorsomedial and ventrolateral cells join to form myotome, and we have the dermatome or dermatomyotome, uh, more dorsally superficial. In D, we have here the skeletal, and here the myotome, and more dorsally and laterally is the dermatome. Here we have completely formed neural tube, and here is the notochord. The lateral somatic frontier is well-defined border between each somite and the parietal layer of the lateral plate is there. And through this lateral somatic frontier, some of the paraaxial mesoder from ventrolateral edge migrate to lateral plate mesoder. So the lateral somatic frontier separates two domains of mesoder, the primaxial domain and the abaxial domain. The primaxial domain, which contains only somatic derived cells, cells derived from the somites or from the paraaxial mesoder only. The abaxial domain is formed by the lateral plate mesoderm in combination with some somatic derived cells that migrate across the lateral somatic frontier. So we have two domains, primaxial domain and the abaxial domain. So we have the primaxial muscle precursor cells and the abaxial muscle precursor cells. And each type of precursor cells gives origin to certain group of muscles in the body. And in this table, you see the different group of muscles coming from which precursor muscles cells, like primaxial and the abaxial. So the primaxial, in the cervical region, it gives scalenes, geniohyoid, and prevertebral. In the thoracoabdominal region, it gives the intercostals. In the upper limbs, it gives rhomboids, levator scapulae, and the latissimus dorsi. In the lower limbs, doesn't give anything. We come to the abaxial, it gives the infrahyoid muscles. In the thoracoabdominal region, it gives pectoralis major, minor, external oblique, internal oblique, transversus abdominis, rectus abdominis. That means abdominal wall muscles, the sternalis, and the pelvic diaphragm. In the upper limbs, it gives distal limb muscles. The abaxial domain gives distal limb muscles and all lower limb muscles. So the abaxial gives the infrahyoid muscles, the abdominal wall muscles, and all lower limb muscles and the distal limb muscles. And we have mentioned that the abaxial contains the lateral plate mesoderm with some somatic derived cells that migrate across the lateral somatic frontier. The primaxial mainly forms the prevertebral, geniohyoid, and the scalenes, the muscles of the back, and the intercostal muscles, and some of the shoulder girdle muscles, the rhomboids, levator scapulae, and the latissimus dorsi. Here in this diagram, in cross section through embryo or transverse section, this is the spinal cord, this is the dorsal part of the embryo, and this is the ventral part. The ventral part will have the epimere, and here is the hypomere, which part the ventral part of muscles called hypomere, and the dorsal part is the epimere. And the epimere supplied by posterior primary ramus or dorsal primary ramus, and the hypomere is supplied by anterior primary ramus or ventral primary ramus. This is the innervation of the muscles. Here, with further development, posterior vertebral muscles, which comes from the epimere, and the hypomere gives a external oblique, internal oblique, and transversus abdominis and the rectus abdominis muscles. All these given by the hypomere. So another classification of the muscles precursor according to the dorsal and ventral rami innervation into epimere, which gives the extensor muscles of the back, and they are innervated by the dorsal primary ramus. And the hypomere, which gives the flexor muscles of the back, the anterior neck muscles, the chest and abdominal muscles, and the limb muscles. All of these are hypomere, and they are innervated by the ventral primary rami. Now let's talk about the limb muscles, and they are derived mainly from abaxial muscle precursors. As I mentioned, all the lower limb muscles and the distal upper limb muscles are derived from the abaxial muscle precursors. The limb bones are derived from the parietal lateral plate mesoderm, parietal layer of lateral plate mesoderm, and the limb buds forms the upper limb from C5 to T1, and the lower limb L1 to S1. This is regarding the somites and the innervation from C5 to T1. This is for the upper limbs, and here from L1 to S1 for the lower limbs. And this includes also the segmental innervation of the skin in the upper limb and the lower limb. An embryo at the week of the 5th week, 5 weeks age, the limb develops and it appears like this in the cranial part and in the caudal part. This is for the upper limb and this is for the lower limb. These limb buds consist of core of mesenchyme. This core of mesenchyme is formed or derived from the parietal layer of the lateral plate mesoderm, and it's covered superficially by ectoderm here and here. Ectoderm at the distal border of the limb bud thickens to form the apical epidermal ridge. At age of 6 weeks, there circular constriction appears distally here in the limb buds that separates the proximal part from the distal part. this constriction. Then at 8 weeks, another constriction appears to separate the proximal part into two parts. So in this manner, the development of each limb proceeds proximal distally, from proximal to distal. Okay, into three compartments. Firstly by appearance of circular constriction, then second constriction. So it develops into three components: the stylopod, which forms the humerus in the femur, the zygopod, and the zygopod formed of radius and ulna, tibia and fibula in the lower limb. And distally, the autopod, which is in the hand, carpals, metacarpals, in the digits, and in the foot, tarsals, metatarsals, and the digits. This is the autopod. So stylopod, zygopod, autopod. Here also stylopod, zygopod, and autopod. The terminal part of the limb forms and becomes flattened, as we see here in 6 weeks embryo, to form the hand and the foot plate. With further development, it gives shape at 8 weeks. Okay, the ectoderm that covers the limb box is called the apical. There it is thickened distally to form the apical epidermal ridge, and you will see in the next slide how the apical epidermal ridge forms the digits or participate in the formation of the digits. The apical epidermal ridge, as we have mentioned, it is a thickening in the ectoderm covering the limb bud, found at the distal part of the limb bud, and by means of apoptosis or programmed cell death, this apical epidermal ridge is separated into 5 parts. These 5 parts result in the formation of the 5 digits in the hand and in the foot. This is an image for human hand. In A, at 48 days embryo, this is a 48-day embryo, we'll see the hand, how it looks like. The cell death or apoptosis in the apical epidermal ridge creates separate, this is the areas, this one, and here, and here, and here, these are the areas of cell death, creates separate ridge for each digit. In B, this is at 51 days, after 3 days from this image, we have the areas of the cell death becomes larger, and the cell death occurs in the interdigital spaces, produces separation of the digits and giving 5 parts of the ridge. In C, at 56 days, you'll see the digits or the digit separation is complete. We have these metacarpals and the phalanges, and the digit separation is complete. This is at 56 days embryo. So this separation is induced by the apical epidermal ridge. The apical epidermal ridge results in induction of the underlying mesenchyme to form the digits. the metacarpals and phalanges, and in the foot the tarsus and the metatarsus, and the phalanges. So all of this process is done through apoptosis or programmed cell death. So how the limbs reach their final position and shape? Firstly, it starts by elongation. It elongates, the limb body elongates. Then segmentation and differentiation of the different bones in the upper limb and in the lower limb. And then rotation, the final step. So elongation of the limb body, differentiation, segmentation and differentiation into different bones. In the upper limb, humerus, radius ulna, and the digits. In the lower limb, femur, tibia and fibula, and the digits and tarsus, metatarsus, and the phalanges of the foot, and then the rotation. In the upper limb, it rotates 90 degrees laterally so that the extensor muscles become posterior and the flexor muscles become anterior, and the thumb is located lateral. In the lower limb, the opposite occurs. It rotates 90 degrees, approximately 90 degrees medially, so that the extensor muscles become lateral or anterior, lateral and anterior, or anterolateral, and the flexor muscles become posterior. And the toe, the big toe, is located medially. This is the normal rotation of the limbs. So the upper limb rotates 90 degrees laterally so that the thumb lies lateral, and in the lower limb rotates approximately 90 degrees medially so that the big toe is located medially, and the extensor muscles are anterolateral and the flexor muscles are posteriorly located. Here in the upper limb, the extensor muscles are finally located posteriorly and the flexors are anteriorly in the upper limbs. So in image A, on the left side, you have a female child with complete absence of the left upper limb. And this is called amelia. Complete absence of one extremity or two is called amelia. Here in this child, the left upper limb is absent. And this is called amelia. On the right side B image, this baby have what is called meromelia or phocomelia. Here there is absence of long bones, the humerus in the upper limb and the femur in the lower limb, and only the hands and feet are attached directly to the trunk. This is called meromelia. Or phocomelia. This is a club foot. The club foot from its shape, the feet are medially rotated. 25% of these cases of club foot are familial and that the prevalence varies across ethnic populations. This suggests that many cases of blood pool occur from genetic causes that result from mutations or microdeletions of certain transcription factors that are involved in hind limb development or learning development. This is called syndactyly, fusion of two digits together, and this is due to abnormality in the process of apoptosis in the apical epidermal region. So this syndactyly with used digits result from failure of apoptosis between the digits. This is polydactyly associated with syndactyly in the left hand. Polydactyly, we have an extra digit 1, 2, 3, 4, 5, 6. Here on the left hand we have syndactyly associated with polydactyly. In this abnormality we see abnormal cleft in the foot here on the right and the left side. This is abnormal cleft in the foot found between the second and the fourth metatarsal bone and the third metatarsal or the third phalanx is almost absent. And this is all related to the apoptosis that occurs in the apical epidermal region of the foot plate. This is what is called Brombelli syndrome. Brombelli syndrome, there is a distended abdomen from atrophy of the abdominal wall musculature. So that the organs are visible and easily palpated because of atrophy of the abdominal wall muscles. This is a Poland syndrome. In Poland syndrome, we have the pectoralis minor, part of the pectoralis major is missing on the patient's right side, and the nucleus is also absent. So this is the end of our lecture. Thank you for listening.